We are happy to welcome you at the online lecture series from the NGS-CN Summer School 2024. As most of you already know, NGS-CN stands for the Next Generation Sequencing Competence Network. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We leave the link in the footnotes, and here is a QR code for your fast access. Welcome aboard, find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
Everyone, for this session after lunch. Hope you got enough energy to follow it and get enough stamina to don't make the siesta after lunch. The aim of this presentation, it's roughly 30 minutes, and I would like to bring you back from the basics of what is RNA, why do we need RNA into a system, toward how do we work with RNA and measure RNA. And I also wanted to show you a bit how do we process RNA data and how do we use them for biological research. So my name is Nicolas Casadei. I'm working at the University Hospital of Tübingen. I'm doing research in neurodegenerative research, animal modeling of Parkinson's disease. And I'm also managing the core facility for sequencing, which is at the University Hospital of Tübingen. So if we think very broadly about the concept, what is transcription? The idea is that inside the context that cells are complex systems that can proliferate and then can play specific functions. They all have the same genomes and different cell types such as hepatocyte or neurons. They are basically very different cells units which originate from the same genomes. So we need to understand that inside the genomes, DNA is a complex structure which can form chromosomes, and we tend to represent genomes by small functional blocks that we call genes, which encode a specific function of an organism. Here, what is obviously important is that not all the genes from the whole genome of an organism, they are not always needed all at the same times. So one of the regulatory mechanisms which is important inside this specific mechanism is a transcription from the gene and the function of DNA towards a transcription toward RNA. And still yet that we can control the gene expression, not the RNA is not always needed everywhere in all the cells. So finally, inside this concept, it's important to see RNA as a messenger, which has multiple functions in the cells and which contribute definitely to the cell function. So seeing RNA as the delivery of the genomic instructions. Typically, if we would look at that with a biological molecule that we are used to as molecular biologists, one would see DNA as these long polymers of different bases, which can be transcribed by a complex mechanism to a messenger RNA. And this trinucleotide of RNA could be then translated to proteins. So the global idea would be that from a DNA, which is static into a cell nuclei, we can get a relatively flexible molecule of RNA, which can be basically translated from the DNA to the RNA, being inside the nucleus, being processed, and could play a role within the nuclei or could navigate outside of the cell nuclei and play a function inside the cell cytoplasm. In order to play this kind of function, we have what we call the post-transcriptional events. And this basically means that from a large sequence of DNA, only specific region would be translated into a pre-mRNA, which contain different region at the beginning or at the end of the RNA molecule, and which contains core region called exomes, which can be basically spliced inside one final molecule of RNA that could be translated into a peptide. But RNA could have different types. They could be different. Until now, we spoke mainly about this messenger RNA, which are basically translating the function of genes within the cells. And they are only representing a very small amount of the total RNA that one could find in a cell, so typically 1 to 5% of it. And these are usually the molecule of RNA which were the most studied because as they directly code for an amino acid sequence of protein, we could understand relatively well their role into a cell or into an organism. But if we think biologically of what is inside a cell and which kind of RNA can we find, most of the RNA are the so-called ribosomal RNA, and they have relatively variable size, and they combine with protein to form ribosomes. So they are RNA which are structural, which play a critical role into the function of the cells, but they don't code for a protein. They are just part of a complex of protein and RNA, which is critical to translate the RNA into proteins. And also a non-negligible part of the RNA are the so-called regulatory RNA, which could be, for example, the tRNA, where that are represented on the right as transfer RNA, or which are also called the small nuclear RNA, which are involved in splicing that I just showed previously. So if we like to see a bit what RNA is doing, how RNA is working, and what is important inside this RNA, one could simplify this RNA molecule being long polymers. And these polymers are based on two different parts. One of the parts is the so-called nucleotide. Nucleotide, if I can show them with my pointer. So they are these parts which are in purple, which are in blue, in red, and in green. These are the variable parts of the RNA. They contain basically the informations, and they have different chemical structures, which allow them to play different functions. And inside this polymer, there is a base that we call a sugar phosphate backbone, which is on the bottom, which basically helps to build up the polymers and which is conserved between all the bases of RNA. Now, what is important to understand biochemically is that if RNA is translated from DNA, they still have different structure and different propriety. DNA, as commonly shown, is a double helix, and it has complementary base pairs. And despite that these base pairs are based on nucleotides which are very similar to the RNA one, there is one major difference is the thymine and uracil, which are two different nucleotides between DNA and RNA, and which is going to play a role later when I'm going to explain you how do we measure RNA and how do we prepare RNA for a quantification. And obviously, another difference between DNA and RNA is the presence of basically OH groups on the molecule of RNA, which is not present on the molecule of DNA. What is important also is because RNA is single-stranded, RNA tends to form hairpins and structures, and these structures they usually play a critical role into the function of the RNA. And one of the most steady structure of RNA is the tRNA, which is used as a transfer RNA to transform an anticodons into a peptide. And the structure of the RNA is very particular because it helps to present in the one hand the anticodon, and in the other hand the amino acid which is necessary in order to build the peptide. So typically one represents RNA being translated to a protein with different tRNA, which can bind on mRNA and which can bind inside the ribosome a growing chain of peptides. So here is a representation of why the molecule of RNA has a specific 3D configuration and why this 3D configuration is important in this case for the function of the RNA. So if we speak now a bit more applied about what is RNA, how do we work with RNA, what is transcription, how do we use transcription in molecular biology, and how do we use it in sequencing centers. We are studying transcription mainly for these 4 different purposes. One of the main one is the gene expression. So we would like to know when are gene being expressed in a specific organism, in a specific tissue, or in specific cells. So we would like to know in which condition is a specific gene upregulated or downregulated. And we would like to study the cascade of signaling, meaning which signal leads to which activation of gene, which reflects which gene expression, and which reflect to which effect inside a cell. Something that is being studied more and more due to the technical development is not only which gene are being expressed, but it's also how are the gene being expressed, meaning which RNA is being expressed when, and how does this RNA is spliced. So that's what we call alternative splicing. And this is the study of how is RNA being spliced, expressed, and how are the different splicing transcript being expressed through a systems. Field where we use also transcription quite a lot is the regulatory RNA, for example, small RNA. They are very important to understand the fine regulation of gene expression, for example, using the study of microRNA, but also the fine regulation of splicing. So what are the mechanisms which are needed in order to change the expression of a specific transcript by another? What does lead to different alternative splicing? Or also like the transport of amino acids. Another field of research in transcription is the study of long non-coding RNA. They play a specific role into RNA-protein interaction, and they are quite well studied in the direction of how does DNA chromatin is regulated, or how specific gene enhancer are regulated, meaning how is DNA configuration being changed by specific long non-coding RNA in order to present a specific DNA 3D conformation, which leads to the transcription of specific genes. So in history, typically methods to quantify RNA started with what we call Northern blot. I don't want to go too much into detail of it, but I think the concept is quite interesting. The idea is if one take a sample, if one isolate the RNA of this specific sample, one can run the RNA through electrophoresis into a gel. One can transfer the RNA, which was via electrophoresis separated by size into a gel. One can transfer it into a membrane, and one can expose this membrane to what we call label probes. Typically, these label probes would be a sequence of RNA of interest that one would like to visualize and quantify on a sample. So if I, for example, design a small probes, which is specific for a RNA of interest for my gene of interest, I can then try to visualize it on an X-ray film. And this method, they were used quite extensively. For example, to validate if a gene was expressed or not in a specific sample. For that, what was done is one would have a control situation, compare it to an experiment, and one would look at a very specific probe of interest, how strong was the signal when compared to an intern gene. the problem of this method is that they are relatively difficult to implement, like the electrophoresis, the blotting, and the staining with the probes. And this probe typically needed to be optimized for every single sequence, making the method relatively low throughput. And the problem of making a gel, blotting it, and staining it usually leads to relatively high variability in the quantification. So this method were very helpful, especially when we were starting to make the first knockouts, validation of knockouts, and so on and so forth. But currently they are rarely used because they are difficult to implement. What is done, what was done later on and is more done nowadays is using PCR in order to quantify, in this case, not RNA, but coding DNA, like cDNA. And the idea there would be to integrate within an amplification of DNA, like coding DNA, for example, using a specific dye, which helps to quantify the amount of DNA, quantify the increasing amount of DNA via a PCR, and use this exponential phase of DNA being amplified in order to quantify the molecule of DNA. This is typically much simpler to implement than a Northern blot. The quantification, despite it's a bit complex, it's very reliable. And we use this quantitative PCR very regularly. For example, COVID-19 was tested using mainly quantitative PCR. The inconvenience of the method is the throughput, because one would need to make a PCR for every single gene that one would like to study. And therefore, it's difficult to study a large amount of a molecule using this specific method. As well, also that one needs to amplify a specific gene. So one needs to know already which region of the genome is interesting to be amplified. When scientists wanted to scale up this method, one of the methods used for scaling up was the so-called microarrays. And these microarrays are a way to detect several hundred or several thousand of specific sequence within one experiment. The idea there is to print on a very small surfaces specific probes which are relatively close to each other. And via a system of microfluidic, one could load a specific sample on these specific regions. And afterward, after hybridizing a specific sample to specific probes, one could quantify how much signal is being done. So if we would have this small array, one could say that, for example, I'm spotting into this different area, different region of DNA or RNA I'm interested to measure to. Can, for example, spot in these three first spots, 3 different regions, which are for one specific gene, and for the next 3, another specific sequence, which is for another genes, and so on and so forth. And then I can compare the signal I get in all these specific dots for all my different gene of interest, and measure a couple of thousands of genes in the same times using one experiment. The microarray were quite successful as a method. What was quite interesting on this method is testing thousands
Recording
of...
in progress.
So looking at thousands of genes at once, one could much better understand mechanisms which happen in a specific systems. But also one could basically scale up this system by, for example, making a microarray for all the transcripts which are known in an organism. And thus one could scale up experiments in different labs using different kind of samples, just by producing in large amounts these microarrays and testing at the whole transcript level all the genes that could be detected and quantify them. The inconvenience so the advantage of the microarray is they get very sensitive, and despite that the analysis is a bit complicated due to the statistics, they are relatively easy to automate. The handling of the experiment is not necessarily very simple when compared to a gel electrophoresis. But this is something that could also be performed in large throughput or even automatized. The main problem of this method is rather than one needs to make a design for this microarray. That means that when starts to process this microarray and try to build them in high throughput, one need to come with a design that cannot be easily changed anymore. And the fact that one cannot change anymore the design of this microarray lead to the fact that, for example, it's difficult to use one array for different organisms. One need one specific microarray per organism, or it's difficult to study a very specific genomic region which was not that well studied before and now is of interest that one would like to use and integrate into this microarray. So despite that the system is still very useful and generate very good data, one see a tendency that they tend to be less used for transcriptomic studies currently in research. So what is being more and more used and what is more now the focus of the rest of the presentation is a method to quantify RNA and gene expression using next generation sequencing. And the concept behind it is not too complicated. The idea is one would take a specific RNA of interest, one would fragment it into small pieces, one would take these small pieces, convert them into a cDNA, and one would sequence these small pieces of cDNA to quantify how many fragments of RNA could be found, but also to study which sequences do this RNA fragment exactly have. So I represented it below. The idea would be from a specific sample, isolating the messenger RNA, taking these small messenger RNA, fragments them in small pieces, convert them into cDNA. The advantage to have DNA instead of RNA is one could use enzymes which are commonly used in molecular biology laboratory. Having cDNA helps us to amplify the nucleotides via polymerase. It also enables us to make, for example, a ligation using enzyme. So there are a lot of advantage to convert a messenger RNA or mRNA molecule to a coding DNA. Once the coding DNA fragments are generated, then the next step is the amplification of them in order to enable sequencing, doing sequencing, and then performing the data analysis. So in the next slide, what I'm going to explain you a bit more is how do we go from a cDNA to a sequencing run. And I'm going to conclude with how do we analyze the result from the RNA sequencing. In transcriptomic, one point which is really important is one need to formulate what do we want to study prior performing the experiment. As I showed you before, RNA are very diverse. We have a structural RNA, we have small RNA, we have long non-coding RNA. So one already need to know what are the species of RNA which are of interest into an experiment. The reason for that is one could already enrich or deplete specific RNA, which might play a function which is of interest in the research or which might be actually just noise that one are not interested in a specific research hypothesis. So here what I did is I represented 4 different conditions. How could be design an RNA experiment in order to detect different kind of RNA. So the very first method I'm showing in A is a total RNA. If we think that the total RNA is a mixture of all the RNAs that we find into cells, one would expect to find inside it a lot of ribosomal RNA because they are the most abundant. One would also find a lot of small RNA, and one would also find a bit of messenger RNA. So here what is written is you have in pink here, this ribosomal RNA, and because they are very, very abundant, a lot of molecules that will be sequenced would be this ribosomal RNA. So you see that it represents over half of all the molecules. Here, what could also be of interest is this green transcript, which are representing messenger RNA. So you can see here basically that they could also be detected via these methods because all the RNA are being sequenced. And what is interesting inside it is if we consider this big square being like the exons and this small line being the introns, couple of molecule would be called spliced, meaning that it will take the beginning of an exon and the end of another exon, but also a couple of reads would end up in what we call introns. These introns could be real introns, or these introns could also be some background DNA that one also process there. Another strategy in order to achieve RNA sequencing would be to deplete all the reads which are into the ribosomes. So if the research question is not targeted toward ribosomal RNA, there is no need to have such an enormous amount of data on the ribosomal RNA. And one of the methods to do that would be to use artificial probes, which are going to decrease this read into the ribosomes. One common method would be, for example, to use specific probes that bind to the ribosomal RNA sequences and which would induce their degradation. So if we would do something like that, as a consequence of it would be that the amount of ribosomal RNA would decrease drastically because that's the aim of the experiment. We would also have still reads which are inside introns or DNA because this method would not reduce them. What we would also have are the mRNA. And what we would also have inside it are, for example, unprocessed RNA or small RNA, for example. The next strategy, which is used a lot in RNA sequencing, is a method which, instead of targeting the ribosomal RNA, is a method which focuses in enriching the so-called poly-A transcript. So what I show very briefly into the introduction is that pre-mRNA need to be spliced in order to become functional and also in order to leave the nucleus of the cells. And inside the processing of the RNA, typically splicing would happen, a cap would be added at the 5' and a poly-A tail is going to be added at the 3'. So one of the methods that we have would be to enrich all the RNA molecule which have a poly-A tail. And by doing that, what would happen is we would get much less read for ribosomal RNA because they don't have a poly-A tail. And the amount of read inside the DNA is going to also be reduced a lot because only DNA region which have a lot of A would be enriched and all the rest would not be enriched. And obviously, all the messenger RNA would be very highly expressed. So this method is really good for removing ribosomal RNA. It's also very good for removing DNA. However, within this method, one would lose, for example, mitochondrial RNA, which don't have a poly-A, or also some small RNA, which also don't have a poly-A tail. And the last method would be to capture cDNA of interest. Here, the idea is to use specific probes for specific RNA of interest and only capture or amplify region of interest. The advantage of this method is that obviously ribosomal RNA, if it's not of interest, would not be amplified. Genomic DNA, if it doesn't share large complementarity to the probes, would also not be enriched. But RNA of interest would be enriched, that these are messenger RNA or small RNA or long non-coding RNA. And a gene, even if it's not very highly expressed because it's targeted, could still be detected. So this is a bit the different strategy we have in transcriptomics in order to target the sequencing of specific RNA of interest, either based on the depletion of ribosomal transcript or based on the enrichment of poly-A transcript, or based on the capture of specific DNA, cDNA of interest. So in this slide, what I want to show you is to explain you briefly what are the strategy we are using to get this specific RNA of interest being sequenced. And this is a very classical method based on a kit. And inside this method, what we consider is one would have already enriched the RNA of interest. So here, this specific representation assumes that an RNA enrichment was done, either based on a mRNA enrichment or a ribosomal RNA depletion or an enrichment. So the very first step to get the RNA sequence would be to fragment the RNA. And the fragmentation of RNA could be done via ions and temperatures. But there are also other methods which could lead to RNA fragmentation. There's the idea to fragment the RNA is because the sequencing method I'm going to present here can only sequence fragment of DNA which have a limited size. So one needs to reduce the messenger RNA, which could be several thousand of base pair, to smaller molecules, which are, for example, a couple of hundred of base pair. Once the RNA is fragmented, what we are going to do is to make what we call the first strand cDNA synthesis. So we are going to use an enzyme to convert our RNA into a coding DNA. Once the first strand cDNA is done, one is going to get readout of the rest of the RNA by doing a second strand cDNA synthesis. So one would end up with a molecule which has two strands of DNA and no more RNA inside it. In this specific protocol, one would clean up all the enzymes that were needed in order to make these conversions, and one would restart with a clean double-stranded cDNA on which the end of the DNA is going to be repaired, and a phosphate is going to be added at the 5'. This is important in order to add what we call the A-tailing, distributing the A bases at the 3' of our DNA molecule of interest, and with this making a ligation of a specific artificial DNA, which is going to be very useful afterward for the amplification of our molecules, but also for their identification. So the idea inside this very specific molecule of artificial DNA is to give them a sequence which help us to distinguish where is the strand, so which strand is at the 5' and at the 3'. And this is going to be important because using a uracil inside these specific adapters is going to basically help us to cleave the molecules and amplify specifically in a direction our molecule of DNA. In order to do that, what we are going to do is we are going to amplify this specific molecule of interest using specific primers. And amplifying the DNA of interest with these specific primers, one is going to be able to add sequences of interest, which are important for the sequencing of the DNA, but also for the visualization of the molecules, meaning that each molecule could be barcoded with a specific sequence, which help us to distinguish which molecule is an original molecule and which molecule could be a result of a deep amplification, meaning that some specific region of DNA, they might be very easy to amplify and they could result to a lot of different molecules that could be sequenced afterward, which are not independent molecules, but just duplicates of the same molecules. At the end of this amplification, one end up with these specific DNA molecules. They contain different signals which are important for sequencing. They contain specific primer needed for the attachments to the NGS systems. They contain so-called barcode, which are important for distinguishing the sample. This is what we call labeling of sample for multiplexing. And they also contain what we call unique molecular identifier, UMI, which help us to distinguish if the molecule are unique or duplicate, and what we call inserts, the region of DNA which are of interest, which represent the RNA sequence. Once these molecules are basically generated, this library preparation of the functional DNA can be cleaned up using beads. So magnetic beads for enrichment of DNA is something that is done extensively in genomics laboratory. And this can be loaded on the NGS sequencer that, if I got it correctly, was presented this morning, like the NGS sequencing, could be amplified on the flow cells, could form specific clusters that could be detected by fluorescence, and generate specific image files which are ready to be scanned by the instrument and be analyzed for bioinformatic analysis. So now what I wanted to show you briefly is how do we analyze this RNA data? How do we check that our RNA was correctly sequenced? And how do we finalize the overview of the data? So one of the very first steps that we are doing is, after the sequencing, we look at all the molecules which are unique and what are all the molecules which are duplicated. And this is, for us in the molecular lab, quite important, that help us to know whether a specific biological sample was sequenced very deep with a lot of new information, or if the sample had a low quality and not much genomic information was retrieved. In this specific case, we can see, for example, that the first three samples had a very low RNA diversity, and most of the reads which were generated are actually duplicate reads, despite that the next two samples have a high biological diversity, and most of the molecules detected are original RNA. What we are also looking at the end with the NGS data is what we call the mean quality score of the base. So every time that we sequence a nucleotide, the system is investigating how good was the signal of the nucleotide in order to decide if the base detected is reliable or not. And what we would see is through the different sequencing cycle, the quality tends to decrease after a certain amount of sequencing cycles. And one could observe is all the samples were sequenced with a good quality, or if they reach a low quality sequencing. What can be done is all the base which have a low quality, they can be removed bioinformatically, and only the base with a good quality can be kept. And what we also typically do is we are looking at the number of molecules which only have an index but no insert. They could be removed. And this would be artifact of the library preparation. And one could also see is how much did we trimmed, how many low quality bases have we removed from the old data. So this is typically what we would do after sequencing to visualize whether sequencing was working well or not. The next step is one would compare the fragment of DNA to the reference genomes and see how much of these fragments are known in the specific genomes or not. Typically, good RNA are going to have high uniquely mapped sequences, meaning that the piece of RNA that we detected, they would be specific only for one location of the genomes, and they would be attributed to one specific genes. It could be that randomly some region could be mapped to different genome location. For many analyses, they would then not be considered anymore. Or it could be that we cannot map them either because the sequence are very short, or because there are some other issues inside it. And what is important is, interestingly, normally most of the molecule, they mapped either in sense or in antisense. And this is due to the fact that when we prepare library, we have a way to define in which direction is the specific molecules. And in this very specific case, the molecules, they tend to be generated in antisense due to the library preparation. One could look, for example, also at if some chromosomes are overrepresented. Typically, for example, if mitochondria is sequenced, for example, using total RNA, they tend to be very highly represented. And finally, what can also look at is where are the fragments of RNA when considering the gene. Ideally, most of the content of the RNA is inside exons. As they could also be inside the 5' UTR, the 3' UTR, so in the untranslated region of the RNA. But it also happens that we get intronic regions. What is quite important is to look at other regions which should not be expressed, for example, intergenic, which could reflect DNA, because a high amount of DNA inside the RNA data would be more difficult to analyze. Finally, one of the last QCs that we do is we are looking at the theoretical size of the insert. As I told you, we need to fragment the RNA for sequencing. And the fragment of RNA, they should have more or less the same size distribution. One could test that bioinformatically. One could look at what we call the saturation of sequencing in order to know, would it be worse to sequence more and more of these samples, or did we reach a saturation and sequencing more won't help to detect more molecules? One could look at where are the reads when considering the beginning and the end of the transcript. Ideally, the reads, they are distributed over all the transcript, and they should not be, for example, a drop of the beginning or the end of the transcript. And finally, there are simulation models which help us to distinguish whether the number of duplicates, so how many molecules were sequenced multiple times, and if they were in a good experiment or in a bad experiment, meaning the genes which are very highly abundant, they should be detected multiple times, and genes which are relatively rare, they should not be duplicated. So the last part I wanted to show you very briefly is what to do with this sequencing data. I told you that in transcriptomics, one of the highlights is detecting gene expression. So if a gene is up regulated, meaning induced in its expression or not, what one would do is one would look at the top 500 genes which are expressed in different samples, and one would look how different are these 500 genes between the samples. So using a principal component analysis, one could see, for example, in this specific experiment that we have 2 wild types versus 3 experiments, that the samples are relatively diverse. However, the wild type samples are a bit more closer than the experiment one. One could represent that in different formats, such as, for example, this correlation plot. And one could see more or less using a different method that the genes which are expressed in the wild types are more correlated than the genes which are expressed in the experiment. Having the genes which are expressed, one could make statistical method in order to see how abundant are the gene, how much difference is being noticed in the gene between the control and the experiment, and what is, based on a statistical test, the probability that they are really different, highlighting the gene that we consider to be dysregulated compared to the one which are not dysregulated. One could plot them into this heat map and put them into different databases such as Enricher in order to detect what do these genes have in common concerning their function or considering which kind of molecule they are. Okay, so I guess based on the time, I'm going to stop there. I hope that I could introduce you a bit to how do we work with RNA, how RNA is prepared for being analyzed, how do we perform transcriptomics using short-read sequencing. I show you the very specific example on how to use the data for the quality control and for the analysis of gene expression. And I also wanted to highlight very briefly that similar analysis could be done also to look at allelic expression, look also at single nucleotide variation at the RNA level, but also to look at different splicing between RNA, or also to see what we call fusion gene, meaning when DNA is modified, we can have RNA which starts on one gene and which end in another gene. So basically genes are fused. And I guess based on that, thank you so much for your attention, and let's get to a few questions.